Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to the 9th webinar in the Women's Health 2024-2025 series. Today's webinar is titled Hormonal and Chromosomal Influences on Autoimmunity and Lupus, featuring Dr. Betty Diamond, Professor of Molecular Medicine at Feinstein Institutes for Medical Research, and Dr. Melissa Cunningham, Associate Professor of Medicine at the Medical University of South Carolina. Today, Betty will present the historical and current understanding of hormonal and chromosomal influences on autoimmunity, and Melissa will follow that up with her work on nuclear hormone action in autoimmunity with special attention to the estrogen receptor alpha. Before we get started, I just wanted to acknowledge our partners for this series, the American Physiological Society, the Society for Women's Health Research, and Scientist.com. We'd also like to say thank you and give a big shout out to all of our series sponsors. None of this would be possible without them. I'm Sarah McFarland from the events team here at Scientist.com, and I'm happy to be your host for today's event. Before I welcome Betty to the floor, I wanted to cover a few housekeeping notes to help you get the most out of your experience today. First, take a look at that interactivity panel at the right of your screen with 4 tabs: chat, Q&A, polls, and handouts. This is where you'll participate in the Q&A for the webinar, as well as access all webinar resources. We want this to be an interactive experience for you, so please send your questions, thoughts, and comments using that Q&A tab, and we'll try to address as many of your questions as we can at the end of the webinar today. You can also explore some links in that Handouts tab to learn a bit more. And lastly, if you're having any technical issues, please try refreshing your browser and ensure that you're using Google Chrome to access this event, as it is the most stable browser. If you refresh and are still having trouble, use that private chat function to communicate with me by sending a chat to Sarah, and I can help you to troubleshoot. And now I'm very pleased to welcome our first speaker, Dr. Betty Diamond. Thank you so much for being here with us today, and take it away whenever you're ready.
Okay, well, it's a pleasure to be here. And what I want to do is just very briefly set some context, provide some context as we begin this session on women and autoimmune diseases. So I just want to remind everybody that autoimmunity is reactivity to self-antigens by T cells or B cells. And some autoimmunity is in fact healthy. So the way we clear cellular debris is by autoantibodies that see apoptotic cells, and they can do this as sort of our green sanitation system, and that's absolutely required for immune homeostasis. But autoimmune disease happens when the autoreactivity is such that there's tissue damage or tissue dysfunction by either T cells or autoantibodies. And when this occurs in the absence of ongoing infection, because we know that during infection, there can be some transient autoreactivity that occurs that then goes away and doesn't lead generally to autoimmune disease. So what we also know is that the antigen receptors on T cells and B cells, the T cell receptor and the B cell receptor, are formed by gene recombination events that occur in the nucleus of a cell in the absence of any knowledge of the world of self-antigen or microbial antigen, so that there are many autoreactive T cell receptors and many autoreactive B cell receptors that get generated by these random events that lead to the formation of the T cell receptor and the B cell receptor. And in the thymus, there is what's called negative selection that eliminates those T cells that are autoreactive. And for B cells, where the maturation of B cells happens in the bone marrow, we know that, in fact, about 70%, 75% of B cells make autoreactive B cell receptors, and there is a process of elimination of these autoreactive B cells as the B cells mature in the bone marrow, and then as they move to the spleen to continue their maturation process. But even with these processes for eliminating autoreactivity, we still know that there are many, many autoimmune diseases. And now there are over 100 autoimmune diseases that have been identified and classified. So autoimmune diseases occur in some 8 to 10% of the population, and they predominantly affect women. We also know that the incidence of autoimmune disease has been rising over the last several decades. This is true for almost all autoimmune diseases. There are many theories about why, but we don't actually know why. But even with changes in the incidence of autoimmune disease, there are still many more women that are affected by autoimmune disease than men. And one of the striking features is that those autoimmune diseases that have the strongest female predominance tend to be autoimmune diseases mediated by autoantibodies. And we know that women make stronger antibody responses to vaccines, and we know that women display more class switching from IgM to IgG than men do. And we know that the autoreactivity that is healthy and important for normal immune homeostasis is IgM mediated, and that IgG autoantibodies are much more likely to be pathogenic. So there are many theories about autoimmune disease, that there's some alteration in either the selection of T cells and B cells during maturation, or in the activation of T cells and B cells, that there is an inappropriate activation of the innate immune response, and that translates into more activation of the adaptive immune response, that there's molecular mimicry with microbial antigens triggering a cross-reactive response that in some people then becomes sustained, that there's poor downregulation of the immune response, or that there is some modification of self-antigen so that it now looks to be foreign to the immune system. So why does this lead to a female predominance? For many years, hormones have been thought to be a major player in this predominance of women in those who are affected by autoimmune diseases. And for sure, as you'll hear, hormones play a role, both the endogenous hormones that we make and endocrine disruptors. And we're now also increasingly aware of chromosomal contribution to autoimmune disease and the incomplete X inactivation that happens on X chromosomes in women. And for sure, infections still remain a very much studied potential contributor to autoimmunity. And over the last decade or so, there are some new contributors that are being looked at. One is stress, one is obesity and altered metabolism, one is exogenous hormones, and one is the microbiome, which can affect autoimmune responses. And with that as a very broad introduction, I'm going to turn it over to Melissa to talk about her work.
Thank you. Thank you for having me as part of this series. I'm going to take a deeper dive on a few of the topics that Dr. Diamond has just mentioned, but again, mainly focus on the role of estrogen receptor in modulating lupus disease expression. So I always like to start with a clinical problem. Lupus, or SLE, is a prototypic chronic autoimmune disorder that commonly affects skin, joints, kidneys, and other organs, but truly can impact just about any part of the body. It's associated with significant morbidity as well as premature mortality. In fact, about a third of deaths from lupus occur in patients who are younger than 45 years old. And despite advances in disease classification systems and monitoring strategies, newer therapies, the survival of lupus patients really hasn't improved much since the 1980s. And nephritis is a particular complication that's feared and is also a source of health inequities that needs to be better addressed. You can see that the 10-year renal survival is only 38% in Black Americans, compared with 68% in white Americans. So we still have a lot of work to do. So the pathophysiology is very complex for this disease, and that's really made the development of therapeutics and clinical trials a challenge. And then the heterogeneity of the disease, as well as really a full lack of understanding of the pathogenic mechanisms, continue to be hurdles to further improving therapies. So as you've heard, or as you know, women are more likely to develop autoimmune diseases. And this epidemiology is pretty striking. So it's been a subject of research interest for many years. And the simple answer is that females initiate a more robust immune response than males do, which helps females fight infectious diseases better than males. But the trade-off is that women are at higher risk for many allergic and chronic autoimmune diseases. But what are the mechanisms behind these immunologic differences? And those turn out to be very complicated to study. So the answer could be viewed as, why are women more vulnerable to developing lupus? Or why are men seemingly protected from developing disease? And you can see how different sex hormones, sex chromosomes, and other things like epigenetics could impact this balance. And those are some of the many things that Dr. Diamond opened our discussion with. So our current understanding suggests that disease onset is triggered by environmental insults, so things like UV light, viral and bacterial infections, smoking, and the list goes on, in genetically susceptible individuals. And the genetic susceptibility in lupus is established. We know that there's an increased prevalence of disease in families, in first-degree relatives, and amongst twins. But we also know, based on genome-wide association studies, that it's really dozens or hundreds or maybe more genes that are all making small contributions towards disease risk in subsets of patients with lupus. And many of these have been identified in genome-wide association studies of other autoimmune conditions, so they're not pathognomonic. So are there links between lupus susceptibility genes and sex hormone receptors? Well, to start, no GWAS to date has provided direct evidence of a polymorphism in estrogen receptor, androgen receptor, progesterone receptor, or other sex hormone receptor, or any of their major regulators like P300 or SRC. There is this ubiquitin conjugating enzyme called UBE2L3 that is linked to lupus susceptibility. And it appears to regulate transcriptional activity of several of the sex hormone receptors in a hormone-dependent manner. So further study on that is ongoing. There is some evidence that there are some different variants of the ER-alpha gene linked to lupus susceptibility or disease severity. But again, these have been small sample sizes and modest p-values and would really need to be confirmed in larger studies. And then another story that's very interesting to do with androgen receptor has to do with the CAG repeats that are linked to autoimmune disease. Specifically, the number of repeats in this particular area inversely correlate with disease activity in lupus and several other disease states. But so far, subsequent studies in lupus and rheumatoid arthritis haven't been able to confirm a clear association with lower androgen activity to support that. So how about epigenetic factors? Well, as you've heard a little bit about AIR, it's a critical regulator of self-tolerance, and it is actually downregulated in females after puberty. One study showed that estradiol downregulated AIR expression in part by increasing CpG methylation of the promoter, and that was what was able to inhibit AIR epigenetically to result in the impacts. There also have been studies looking at global DNA methylation changes during puberty, many of which are near predicted estrogen responsive genes, and are indeed enriched for immune processes. And then some more specific pathways, this one out of the Sokos lab, they showed that estrogen induced DNA methylation via CREM alpha and DNA methyltransferases in T cells. So again, still work ongoing on some of these more specific things, but you can see lots and lots of study on different aspects that are impacting the immune system in different ways that relate to biologic sex. So getting into the sex chromosomes, the sex chromosomes themselves are almost certainly involved. And this really makes sense because the X chromosome contains more than 1,000 genes in comparison with the Y chromosome with only about 100. And many of those genes relate to the immune system. And you can see some of them outlined there. I'll mention TLR7 because that has known gene dosage effects in lupus. And so you can imagine how if you have dysregulated X chromosome inactivation, a double dose of some of these genes could certainly predispose to autoimmunity. And in fact, TLR7 has been shown to escape X inactivation in myeloid cells and B cells. So these studies are ongoing, and we now also have some additional data that exist, which is the major long non-coding RNA that performs differential X inactivation is also perhaps a direct activator of toll-like receptor 7. So more to come on that. The epidemiology has also provided support for the role of the X chromosome for many years. So in addition to the fact that the female bias is observed before puberty, so in lupus, so childhood onset lupus is still 2 to 3 times more common in females, even before puberty. And then we also see a similar lupus susceptibility in Klinefelter men. So these are men with 47XXY, so an additional X chromosome. And there's also an increased risk of lupus and other autoimmune diseases in trisomy X women. So I think the influence of the X chromosome dose on lupus disease risk is established, but the mechanisms are not entirely clear yet. So is that the whole story? Well, as I've already alluded to, genetics, epigenetics, X chromosome inactivation, etc., are not the whole story. Of the many risk factors that we've already mentioned today, including genes and environmental exposures and XCI, none of those explain the steep rise in incidence of lupus at the time of puberty that only happens in females when they become reproductively active. And so we also know that hormones can influence disease activity, and there's a long line of evidence linking hormonal status and immune function in both healthy and affected individuals. And so estrogen, among other sex hormones, has been implicated in immune function. But estrogen in particular is very complicated. Estradiol, which is the main type of estrogen that circulates when females are premenopausal and not pregnant, often works in connection or in concert with other hormones for a particular function. It can also compete or be antagonistic, and all of that depends on the cell or tissue type. So consistent with this, estradiol is immunostimulatory in some diseases and inhibitory in others. So for example, estradiol is protective in autoimmune encephalitis, which is a multiple sclerosis model. And it also ameliorates collagen-induced arthritis, which is a rheumatoid arthritis model. But in contrast, estradiol is generally pro-inflammatory in lupus models. For example, supports autoreactive B cells. So when we think about sex hormones, while some of them, like androgens and progestins, are generally immunosuppressing and prolactin is immunostimulating, the answer for estrogen is that it really depends on the cellular milieu. So much more complicated. But if we can better understand its tissue-specific mechanisms, we may be able to harness that knowledge for its therapeutic value. So the piece of the puzzle that I study is the estrogen receptor action, specifically ER alpha. And that came from when I joined my mentor's lab, and they had just published this study where they had crossed an estrogen receptor alpha knockout mouse strain onto a lupus mouse strain and found that the female mice were profoundly protected. So not only did they survive longer, but they had much better kidney disease, so lower renal pathology scores, urea nitrogen, reduced proteinuria. If you can see my arrow, this is the proteinuria that's reduced to near normal levels compared to the wild types in hets. And this was despite them having actually higher autoantibody levels. So in this case, higher double-stranded DNA levels. And so you can see that here in the knockouts, and immune complex deposition in the glomeruli of the kidneys of those animals was also not different, despite the knockouts being profoundly protected. So we reasoned that this protective phenotype was distal to the break intolerance, distal to antibody production, autoantibody production, and immune complex deposition. And so maybe this was locally protective, the ER deficiency, and reducing sort of a vicious cycle of inflammation that occurs as a result of autoantibody production and immune complex deposition. And so, and this was also not strain specific. We showed the same thing in another lupus strain, MRL-LPR. So I wanted to study immune mechanisms of estrogen receptors. And just to kind of further convince you of their importance, I like to show this, this phylogenetic analysis, which basically shows that that estrogen receptor is the common ancestor for all steroid hormones, goes back 600 to 800 million years. And so given the ancient nature and the widespread expression and role for ERs in physiology and pathophysiology, it's not surprising that estrogen is implicated in the development of many diseases, not just important in reproduction. So cancers, cardiovascular disease, neurodegenerative diseases, and certainly autoimmunity. Another set of ancient receptors I've kind of already mentioned, toll-like receptor 7, lots of evidence that that's an important driver of inflammation in lupus, and I'll be talking more about that. It also contributes to enhanced autoimmune responses in females more so than males, and more recently has been confirmed to be relevant in human disease. Our laboratory has shown that TLR-induced inflammatory cytokine production is actually blunted in the absence of ER alpha. And that's in vitro, and I'm going to show you some studies about why that took us down this path. We thought this could be very straightforward, that maybe estrogen receptor alpha has a role for, it is required for robust TLR7 induced inflammation. But around that time, my project was taking a different turn because we learned that the mutant mouse model we had, that was what most people used as an ER alpha knockout starting in the 90s, was actually not a null mutant, but in fact an AF1 mutant. And so this is the major transactivating domain for ER alpha 66, which is the classic ER alpha. And you can see that the ERCO that was made by NIEHS in the 90s was missing this. So certainly was a functional knockout, particularly in reproductive tissues where AF1 is required for hormone activation. But because this mouse was still expressing, a short version of this receptor that included the DNA binding domain, the ligand binding domain, and an additional AF2 activation domain, we were interested to see if that was going to have a different phenotype than a true null. And I also just want to point out that our original mutant also expressed this endogenous variant, the 46 kilodalton version. So this short version that endogenously does not have the AF1 activation domain. So we went and back-crossed these true ER alpha deficient mice to our lupus mice. And we were sort of surprised to see these are actually data that are the same, but I think it's a little bit easier to look at the graph on the right. And so we also did ovarectomies on these mice to control for the hypergonadism that happens when you knock out ER alpha. You're basically knocking out a feedback loop. And so you get very high levels of estrogen and testosterone. So when we ovex these animals, the original unmanipulated animals were indeed still protected. But as soon as we removed the sex hormones, regardless of the fact that they were deficient, they were no longer protected. And in fact, the animals that were repleted with estrogen were, if anything, worse. And so we thought this was interesting, and we have hypothesized that this is probably due to the high levels of testosterone that exist in those unmanipulated mice, similar to what we saw in our ERCO shorts, or the original ERCO mice. And so we wanted to then, before I do that, let me just show you the kidney disease again, just to prove to you that ER alpha deficiency is not protective. You can see these sclerotic looking glomeruli that are no better in the ER alpha deficient mice, regardless of whether you give them back estrogen or not. In fact, if anything, the renal pathology is worse in the animals that do not express ER alpha, actually suggesting some of the negative impacts of estradiol may not even be ER alpha mediated. So moving on to autoantibody levels in those animals, ER alpha deficiency was also not protective against autoimmunity. So just like in the original study, regardless of ovarectomy or estradiol repletion, the deficient mice still had high levels of anti-double-stranded DNA. So we were not protecting the animals regardless of testosterone by eliminating autoantibody levels. So in parallel, we wanted to do the similar studies in our original mice because we thought, well, maybe all of this was just a testosterone effect. But when we went back to those mice, we were very surprised to find, and again, these are similar data on the right and left, but a little easier to look on the right. We were very, very surprised to find that in this case, the animals that were ovarectomized and were carrying this AF1 mutant, what we could call the ERCO short, were indeed protected even if they were ovarectomized. And in fact, the only way they were ovarectomized, the only way they were protected is if they also were given back estradiol. So now we knew that this effect required estradiol, and something was going on that was active to do with this and didn't have to do with just being deficient in the ER alpha. And these are the renal scores from those mice, again, very much protected in terms of proteinuria. Their renal pathology scores were completely normalized almost. And you can see this was not true in the ER alpha deficient mice and not true in the ERCO short mice that were not given back ligand. Again, similar anti-double-stranded DNA levels here. You can see in the bottom left. And this was not impacted. The testosterone was not required for this protective phenotype. As you can see, the animals were all overreactomized. We did not replace testosterone. And still, these ERCO short mice were the ones that had near normal glomeruli and even a reduced immune complex deposition, despite having serum autoantibody levels that were similar to wild type. So we went to look at the inflammatory infiltrate in the kidneys of these animals by flow cytometry. And again, you can see in the middle here that the ERCO short animals that are given estradiol have a reduced inflammatory infiltrate. Here we're looking at myeloid cells, C11C, C11B positive cells. But this is also true of B cells and T cells, that they were significantly reduced in the kidneys of the protected animals. We were very interested in the myeloid compartment at that time because there were other groups that were showing the importance of ER alpha in the development of different subsets of dendritic cells and macrophage. And we did, in fact, find that there was an altered ratio of inflammatory DCs to tolerogenic DCs. And that was mainly due to a reduction in CD11 positivity in those. So you can look here and see that the protected animals have a reduced CD11B positivity on these MHC2 positive, CD11C positive, likely to be DCs because of high CD11C positivity. These are reduced, but the CD8 positivity, which suggests tolerogenic DCs, is not impacted in the same way. In fact, if anything, it's increased over the null mice. So now we understood that while ER alpha deficiency did not provide protection, the ER alpha short mice that we had originally studied were in fact protected, even if testosterone was accounted for. And what they did need was estradiol repletion, which makes sense because they still have their ligand binding domain and a DNA binding domain, and this AF2. But we were wondering, is it the lack of AF1, the imbalance of these two, or is it just having kind of a double copy of this ER alpha 46? And I'll show you some reasons why we think that might be important. So just a little bit about ER alpha 46. This is a short variant of ER alpha that only weakly mediates transcription responses to estrogen, mainly because most classic hormonal effects require that AF1 domain. However, because it retains its AF2 function, and it also retains the areas that are responsible for cofactor and SERM binding, we are wondering whether or not this is acting as either a dominant negative or having its own membrane or genomic action, and that's what we are currently trying to study. So a working hypothesis would be that while classic ER alpha is going to, bind ligand and dimerize and move into the nucleus, ER alpha 46 may be able to bind DNA more tightly. It actually has been shown to have higher affinity in some cell types for DNA, and has no AF1 function. So in any cell type where AF1 is required for transcriptional activation, you are going to see that potentially blocked. Another possibility is that ER alpha 66 can also tether to other transcription factors like NF-kappa B, and it can bring in co-activators like P300 and SARC as part of that. But ER alpha 46 has been shown to recruit co-repressors, so things like SMART and NCOR, which could potentially turn off pro-inflammatory genes. And so one of the ways we're trying to understand the mechanism of this is by overexpressing 46 and 66 in dendritic cells. And so we're using, and some B cells as well. We're using electroporation, and we've done some pilot studies to show that this does impact inflammatory genes. So you can see that when 46 is expressed by itself, there are a number of inflammatory genes that are downregulated. It doesn't appear to compete at least 1-to-1 with 66. So when they're both there, 66 kind of wins out. Well, we are now starting to do ratio studies where we're increasing the amounts of 46 over 66 to see if it does have that ability at a certain level. And this is just showing you one of those inflammatory genes, IL-1 alpha, which has been shown in our mouse models and cell systems to be an important TLR7 downstream endpoint of TLR7 inflammation. And you can see that when 46 is overexpressed in these dendritic cells, there is a 40-fold reduction in IL-1 alpha message. And that actually doesn't change even with TLR7 agonism. So again, still trying to work out mechanisms of how 46 is having its effects. Moving forward, we do also have a human project. We've just completed recruitment of 60 patient samples for digital drop PCR, and we are investigating whether ER alpha endogenous 46 and 66 ER alphas are differentially expressed in lupus patients versus healthy controls. We're specifically looking at monocytes, and I'll show you in a little bit why we also think that is important. So the relevance to human disease in immune cells for ER alpha 46 and in lupus is still to be determined. And then I wanted to talk a little bit about membrane action. This is another major project in our laboratory. It's been known for a long time that ER alpha can be adjacent to or within the membrane, and rather than having genomic mechanisms, it has non-genomic or rapid extranuclear signals that result in transcriptional responses. And so this is one of the things that we also think could modulate immune function. And again, just the model for that is, there's lots of classic actions of ER at the EREs, but some of these things, some of the estradiol responses we see are too rapid to be explained by genomic mechanisms. And so we are trying to study some of these effects in immune cells where ERs are basically only at the membrane. And the way we're doing that is with mice that express ER alpha either only in the membrane or only in the nucleus. And so this one's pretty straightforward. It is, let me go backwards, sorry. It's a point mutation at this site, which requires, basically this precludes palmitylation. If you don't palmitylate the protein, you can't traffic it to the membrane. And then this is a little bit more difficult. The E domain has been shown to be the only domain that's required for membrane signaling. So we've got these mice from our collaborators. You can see the E domain still has a ligand binding domain in the AF2, but it doesn't have any genomic or nuclear functions. And you can see their expression levels here. And so when we first got these animals, I had a college student summer student do a very simple experiment just to look at spleen cells taken from these mice. And in ovary intact mice that were stimulated with loxorubine, which is a toll-like receptor 7 agonist, you could see that the mice expressing membrane-only ER had much higher levels of IL-6 produced from them. However, we also quickly were phenotyping these animals and found that much like our other ER alpha mutants, the membrane-only ER had hypergonadism, so very high levels of testosterone and estradiol that have to be corrected in order to study them. So we went on to do the castration studies. And here was interesting. We found that there actually was no difference in the nuclear-only ER from wild type when they still stimulated just as well as the wild type, whereas the membrane ER expressing animals did not. And if we correct that, there seems to be sort of no difference. So what this did suggest is that membrane ER alpha does require ligand for its effects, but it appears that nuclear ER does not. So we are continuing to study that, but we've taken this now into our animal models. Right now, we've just done an inducible animal model, which is not perfect for lupus prone, but it does represent lupus-like disease. So I'll show some of this data. So these are still on B6 background, and we're looking at spleen cells. And here you can see that these mice were treated with a TLR7 agonist, R848, to induce lupus-like disease. And this is the treatment strategy down here if you want to look at it. And so you can see that we and others have shown they get splenomegaly. That's expected. When we look at CD3 or T cells, so these are just basic markers of spleen peripheral subsets, there were no differences either in the treated group or amongst genotypes, whereas B cells were induced with the R848 treatment, but again, maybe a trend towards increase in the mowers, but not specific difference between the genotypes. That was also true of CD11B positive cells, so enriching for macrophage, similar enriching for CD11C positive cells in the spleen. However, when we looked at monocytes, there were very clear differences. It looked like membrane ER was required to some extent to have a normal response to R848 in terms of the absolute number of cells developed. And that didn't appear to be true with the neutrophils, so the Ly6C positive, Ly6G positive cells. So we went on to look at bone marrow, and we can see that there is the increase to some extent over nuclear only with the membrane only, and that appears to be mainly CD11B positive cells. So we are seeing this in both the vehicle and the stimulated cells. So something that membrane ER is mediating in the bone marrow. We saw the same thing with Ly6C, and of course the same thing with the double positive cells. So we are focusing more now on inflammatory monocytes, even though we did see some differences in CD11C cells as well from the bone marrow. So those seem to be different with regard to their development versus their peripheral involvement in the spleen. And then just to show you, B cells were not impacted genotype-wise. So again, they are, just like in the spleen, they are increased in the bone marrow when stimulating with TLR7 and creating this lupus-like environment. But there's not a difference if anything, there's a trend towards nuclear ER alpha being more important or being more responsive to TLR7 in stimulating B cells from the bone marrow. So to date, membrane and nuclear localization of ER alpha appears to differentially impact the development and maybe function of immune subsets. The largest impacts seem to be seen in the myeloid compartment, particularly on inflammatory monocytes. And we have now back-crossed these mice onto a lupus-prone strain, so the B6MBA2 mice, and we are studying their disease phenotype, particularly with regard to kidney and cardiovascular disease, as well as their immunophenotype. So just in the last 2 or 3 slides before we end, I wanted to talk about one final piece that we are very interested in our lab, especially as a potential therapeutic to modulate estrogen receptor activity, which is selective estrogen responsive modulators, so SERMs. And I want to specifically focus on PAPI. So SERMs, selective estrogen receptor modulators, with our next generation SERMs, that there are many, many of these, especially from the breast cancer and oncology world, that we can kind of steal and borrow and test. They have comparable binding affinity for estrogen receptor as estradiol, so they compete well. Many of them have anti-inflammatory effects, which makes them attractive to us. They can be tissue selective, so they don't impact reproductive tissue or fertility, which is critical for our patients who are, many of whom are young women. They're also useful, as I said, for studying non-genomic effects of estradiol. And mostly to date, they've been studied in breast cancer cells, and very little is known about their effects in immune cells. So this is something that we are interested in, particularly this PAPI, which has been not studied very much in immune cells yet, studied mainly in metabolic and vascular tissues, where it works by disassociating very quickly. So it binds and dissociates. So it's never able to basically affect transcription because it doesn't stick around long enough to do that. But it can still activate kinase cascades and has been shown to be active with membrane ER in activating mTOR and MAP kinase pathways. And so our working hypothesis is that perhaps PAPI, which acts through membrane ER alpha, will suppress TLR7 mediated production of pro-inflammatory cytokines and have fewer or minimal genomic side effects. Certainly in reproductive tissue would be the hope. And so in our pilot studies, you can see that PAPI alone does not increase IL-6 message. Here's the TLR7 stimulation of IL-6, and you can see that the addition of PAPI reduces in a dose-dependent way IL-6 production. And we've looked at a few other message levels. Again, this is looking at short stimulations with TLR7 agonism, and we can see reduction in TNF alpha and MCP1 and IL-1. And these experiments were done in primary cells from mice. So we are taking these into preclinical studies, and we'll hopefully have some interest at some point in trying these in humans. So are SERMs a potential adjuvant therapy for lupus? We're trying to build on the foundation of prior studies looking at DHEA, which can be turned into testosterone and estradiol, but also estrogen blockers, which all showed modest improvements in lupus disease activity and quality of life, but the effects were really, they were inconsistent, they were subtle, and the side effects were really unpalatable. So we need targeted, like many of our therapies, we really need targeted treatments. But again, there's a lot of interest in this because in both mice and humans, there's very clear evidence that endogenous estradiol has many protective effects on bone integrity, on brain function, on vascular health. So there remains a great interest in harnessing the beneficial effects of estradiol, and tissue-targeted SERMs might be one way to do that. So these couple of SERMs that we're studying are already commercially available. They have a very unique mechanism of action, and preliminary evidence suggests anti-inflammatory effects. So we'll be, like I said, using these in our lupus models. So in summary, despite studies on estradiol signaling and its effects on different immune cell types in lupus over the years, our understanding of ER mechanisms of action in both even normal immune function, but certainly autoimmunity, really still remains incomplete, probably because it's so complex and does so many different things. No current therapy addresses the role of hormones in lupus. It's a very complex biology. There's not going to be one answer. Estrogen is not bad. Testosterone is not good. We'll need to work. This is going to work to our advantage as we develop these more targeted therapies and next generation SERMs, because hopefully we'll be able to tweak things rather than just blast one part of the biology. So I think the future is bright for looking at the effects of both sex hormones and nuclear hormone receptor action in immune cells. We have a lot of new tools, lots of new sequencing tools, CRISPR tools, to better understand how these nuclear hormone receptors act and what their interplay is. And with that, I will join Dr. Diamond and take any questions.
Thank you so much, Melissa, for that fantastic presentation. And also thank you to Betty. Really appreciate you guys being here, and great work that you're putting out. I'm going to just take this moment to let you catch your breath and get a sip of water. And I'm going to also remind the audience that we do have that Q&A panel. So any questions that you do have for Dr. Diamond and Dr. Cunningham, please submit them in that Q&A panel, and we'll get to as many of them as we can today in about the next 10 minutes. And lastly, I did want to remind the audience again, if you have to leave early, we do have a survey for you to complete. And that should be appearing on your screen right now. So like I said, if you have to pop out early, please just give us your feedback on your experience today. We really appreciate that. Okay, and so I'm going to bring Melissa and Betty back on here. I'm just trying to get Betty's camera going. There we go. Perfect. Okay. If you guys are ready, I'm going to get started with our first question.
Perfect.
All right. I think this one is directed at you, Melissa. The first question is, how does estrogen-based contraceptives affect lupus patients of reproductive age?
Okay, so this has actually been, I know Dr. Diamond can certainly answer this as well, but this has been studied in the Selena trials. And so what the general answer is that if patients don't have antiphospholipid antibody and other clotting disorders, in general, OCPs are safe, although they have, there's sort of a hierarchy of how we like to use them. There does seem to be a mild flare rate in the use of hormone replacement therapy in patients who are postmenopausal. But again, those flares were generally mild. So those medicines are not contraindicated, but we still tend to avoid them. And they do have some impact, but they don't seem to cause, don't necessarily seem to unmask disease or cause severe flares. I'll let Dr. Diamond answer.
Can I add to that?
Yes, please do.
I think the way the study was done, the clinical trial was done, determined the outcome. Because in the hormone receptor and the hormone replacement therapy, all comers were included. And in the oral contraception study, only those who had stable disease for a number of months prior to enrollment. And if you're menstruating, if you're cycling, and you have a hormone responsive disease, the likelihood that you will be stable over a number of months is much less. And so I think that by insisting appropriately that people have stable disease, what they did was not include in the trial those people in whom estrogen might actually precipitate the flares. So I don't know that, but I think we should be cautious in interpreting that study to say that oral contraception is completely safe in all individuals with lupus. Of course, oral contraception has changed a lot since then. And those were the days when there was a lot of estrogen there, which isn't the case anymore. So it's sort of moot in terms of using oral contraception.
Absolutely. There's a follow-up question here on that, on progesterone and non-estrogen based contraceptives. Can you also comment on those?
Yeah, so so I don't study these things in the laboratory, but as a clinician, I use, and this is really straightforward, there's a one-page hop step from Duke that sort of takes you through how you choose medicines. And as Dr. Diamond said, very important that as you choose anything, you're sort of thinking about the activity level of the patient and their risks based on clotting, so if they have antiphospholipid antibodies or not. But in general, we use like mini pill and IUDs and those kinds of things that are non-estrogen based in most of our patients, but particularly if they have activity or have APLs.
Absolutely. Betty, did you want to add to that?
Oh, I think that's absolutely correct.
That covered it. Awesome. Thank you. Next question here. Based on your presentations today, how might this understanding of hormonal and chromosomal influences on autoimmunity inform future treatment strategies, especially in terms of targeting hormonal pathways?
Well, there's still a lot to know about. So at the risk of showing my cards, I'm sort of interested in whether or not X chromosome inactivation and what happens with X chromosome is impacted at puberty or impacted by the things that happen when this is so by sex hormones like estrogen. Is that changing? So the only way to know that is if we studied pre-puberty and post-puberty lupus patients, samples, things like that, to see sort of what happens with XCI. In terms of treatment strategies, I haven't gotten that far. I don't know if Betty's been thinking about this at all. I mean, I'm very focused on the idea of tweaking the sex hormones versus how we might. But we need to know the mechanisms of how XCI is regulated before we could really think about therapies.
Yeah, I think regulating the sex hormones, we have a lot more tools in our armamentarium now. I think these are very attractive approaches because in a disease like lupus, it's not in and of itself very immunosuppressive, yet a very high percentage of our patients die of infection. And so the immunosuppressive therapies we use now are way too immunosuppressive. And I think tweaking hormones conceptually is a non-immunosuppressive way to think about therapies. And I think what we need is not just more effective therapies in lupus, but much less toxic, and that means primarily immunosuppressive therapies.
Absolutely. Yeah, well-tolerated therapy is the number one goal. We've got lots of questions coming in, so I'm going to keep going. A few people have asked, I'll paraphrase, what influence does menopause have on lupus disease activity?
So yeah, I'm going to answer this more as a clinician. I have seen some patients, and not just with lupus, but with other autoimmune diseases, where menopause, their disease just keeps coming. But in general, it does seem like many patients remit. But I don't know that we've ever really studied this in a way that would allow us to answer that question, to say that, when what we know is incidence changes for sure. Like, we're not diagnosing menopausal women with disease in the same way that we diagnose women in their reproductive years. That's certainly the point at which they are most at risk for incidence of disease. But in terms of people who already have disease, I think, as Betty said, that my biggest concern with people as they age is just waning immune system in general, and our toxic therapies, and trying to back down. So if they, I'm really trying to do less is more at that point. But I don't know of any data that tells us that menopause does something different in terms of disease activity. Are you aware of that?
So, I would just add one thing to what Melissa said, which is there are studies that suggest that over the past decades, while the incidence is not nearly so high in menopausal women as it is in younger women, that it is increasing. So when I talked about the incidence of autoimmune disease generally increasing, it's also moved into an older age group. Generally, it's not so severe in that age group, but and why that is, again, we don't really know.
Yeah, I would say that those who are I would agree that they're not as severe. They may meet criteria, but they're not the same subset of patients who are very sort of ill vulnerable populations in their 20s.
Absolutely. Thank you both for commenting on that question. We have 1 minute left. I'm going to just ask the last questions. We're going to go a little bit over here. This person has asked, one of the big differences between females and males is increased high molecular weight adiponectin, which has a Q1 complement domain and collagen domain, seeming to coat bacterial insults in collagen. Ooh, okay. Do you see differences in adiponectin expression and function between different mouse genetics and lupus states? So, Melissa, I think that one's for you for now.
I don't know. I'm going to let Dr. Diamond have. I don't know too much about it.
I don't know that it's been looked at in lupus. It's been looked at in diabetes and mouse models of diabetes, but I really don't know. So I'm not sure that I can answer that. And when I think of the different sort of transcriptional profiles that have been looked at, they really wouldn't be including all the cells that make it different. So
most likely unknown.
No.
Yeah.
Awesome. Well, it's something to look at in the
But
future.
it is important. I do think it's important to think of cells and molecules that we don't ordinarily think of as immune cells as potential mediators or potentiators of autoimmune diseases. And I think one of the other things that we don't study enough, because it's harder to study, at least in humans, because the tissue is less accessible, is why some people might have a kidney that gets lupus nephritis and the same antibodies in a different person might not cause lupus nephritis. So I think the question is asking us to have a broader vision of these diseases, and I think that's really appropriate.
Mm-hmm. I would agree 100% with that.
Absolutely. You guys are in a very elusive, mysterious field. So who knows?
Work for a while.
But important one.
Super important. Like I said, that's going to be the last question for today. I want to thank you both so much for your time, your expertise, and for sharing your thoughts and your experience in our Q&A today. It really was a pleasure to have you with us, and I hope that you had fun.
Thank you.
Thank you.
Okay. And to our audience, we really appreciate you being here live. We know you have busy schedules, so having you here with us today is really important to us. And we hope that you enjoyed your experience today. Please join us for the next event in this series next week on Wednesday, February 5th at 11 a.m. Eastern Time. You'll receive a reminder email with the link to log in for that, so make sure you block it off in your calendars. A quick reminder that a recording of today's event will be available soon, so keep an eye out for an email giving you access to that. Additionally, a survey will appear at the end of this webinar. If you didn't already complete it earlier with the pop-out link, we'd love to get your feedback on your experience today. We do read all of your comments, so we'd also love to know what webinar topics you'd like to see in the future. And finally, if you have questions for Betty and Melissa, you can submit them in that survey as well. In closing, we hope that you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society, and we will see you next week. Thanks, everyone.